the next question we ask are, what immune cells are infiltrating the tumors? So in projects such as TCGA, when they were developed, there are pathologists looking at the tumor to ensure that the tumor has over 50% purity. Because when you do DNA or RNA sequencing, you want to make sure you are really sequencing the tumor and not really sequencing a lot of the normal. And this is in the early days of TCGA. You think that tumor with high purity is really, really important. But later on, scientists realized that a lot of these normal cells are not necessarily, for example, you don't want to sequence normal breast epithelial cell. You want to sequence the malignant epithelial cells. But a lot of the normal cells here are not necessarily normal epithelial cells. They could be immune cells, T cells, B cells, dendritic cells, fibroblasts in the tumor, which are actually quite important. And so especially in the tumors, the very good immune cells that can kill the cancer cells are T cells and NK cells that are key factors for patient survival and immunotherapy efficacy. And so pathologists are now learning to stain for this immune cell infiltration. They are also called tumor infiltrating lymphocytes. And so you can see this is one tumor where the tumor immune infiltration is low. This is less than 30%. This is staining for immune cells. And this is what we call immune hot tumors, or tumors with abundant infiltrating lymphocytes. And so this is from pathology, but recently there are also algorithms that can predict immune cell infiltration in the tumors based on RNA sequencing data. So there are two different approaches for this. One is a gene set enrichment type of approach. Basically, if we know what genes are uniquely expressed in T cells, what genes are uniquely expressed in B cells, what genes are uniquely expressed in macrophage, we can create those gene signatures. We say, okay, these 50 genes, these 20 genes, we create a signature, and we use this signature to evaluate each tumor to say, in this tumor bulk RNA-seq, do I see a high signature for B cells? Do I see a high signature for T cells? And so that will kind of give us kind of a relative estimation of immune cell signatures across different tumor types or tumor samples. And so this is one approach. They are using kind of a gene set enrichment analysis to evaluate the immune cell infiltration. The other type of algorithm is an immune deconvolution approach. So if previously people have done a sorted immune cell expression profile, we might have information of how dendritic cells, it is like a sorted dendritic cell, we have their genome-wide expression. And this is kind of a, say, T cells or maybe B cells, you can get their expression. And you can either use all the genes, or usually people pick out still, maybe tens or hundreds of genes that are kind of uniquely expressed in those immune cells, and then do immune deconvolution. So basically, you think that the final tumor expression is the different immune cells that are mixed at different levels, which will give us the final expression level of all these genes. And so the immune deconvolution method is to ask, okay, if T cells express these genes at this level, dendritic cell express these genes at this level, macrophage express these genes at different level, what level, what infiltration level can I estimate? This fraction or the estimation of the abundance is what you try to do. You estimate this in order so that at this proportion of T cells, this proportion of dendritic cell, or this proportion of macrophage, you will have the final expression that's observed on the tumor. And so this is a deconvolution type of approach. There are also many algorithms developed And so far, analyzing a lot of different TCGA tumors, it seems that immune cell infiltration really have very strong influence on patient survival. For example, if the tumor has overall very high level of macrophage infiltration, in here, each column is a cancer type, and each row is one major immune cell infiltration level. And the little square indicates the survival death risk of the patient. And red means bad, blue means good. And if the square is big, it means the p-value is more significant. And so you can see here across many cancer types, overall, this is very red, even though only a subset of those are statistically significant. But what it says is that when the tumor have overall higher macrophage infiltration, patients usually don't do so well. But if they have a very high level of CD8 T cells, you can see in this row, it's mostly blue. If the tumor has overall higher CD8 T cell infiltration, they usually do better. And so that's kind of a useful thing, based on the tumor, whether you can check on the tissue slides or based on the RNA sequencing, you can already make some guesses of how well the patient will survive. Recently, we developed a computational algorithm called Timer. We have our own algorithm, which is a deconvolution method, but we also incorporated 5 other immune deconvolution type of algorithms together to evaluate all of TCGA. So for example, if you want to ask how is antigen presentation associated with CD8 T cell infiltration, so the Timer algorithm has already looked at the expression level of the genes and also the immune infiltration of the different tumors in TCGA. And so here now each row is one cancer type and each column is a immune infiltration estimation methods. For example, there's a Timer estimate, there's EPIC estimate, and Excel is a different algorithm, QuantiSeq is a different algorithm, but you can see red means positive correlation and blue means negative correlation. So from this, you can see overall, when the tumor has higher B2M gene expression, the tumor overall have higher CD8 T cell infiltration because you see the expression of the gene and the CD8 T cell infiltration are positively correlated in many cancer types, regardless of what algorithm we're using to make the prediction. And so this kind of give you some idea what genes are really important in helping in T cell infiltrating into the tumors, right? So this is a useful thing. Recently, there are also single cell techniques such as single cell RNA-seq. You can also use this to estimate the immune infiltration. And so if you remember from the single cell RNA-seq part of the lecture, in the tumors, there might be malignant cells, but there also can be T cells and B cells, and you can compare this. So there are some important factors to know. First, it's okay to compare the same population across different samples. Say, if you want to ask, overall, when we look at CD8 T cells, you can ask which samples have higher CD8 T cells. But then we have to be a little bit careful when we compare different cell populations in the same sample. For example, you can't really compare the absolute level of neutrophils versus T cells, because for some reason, neutrophil is very difficult to be prepared into a single cell RNA-seq reaction. A lot of neutrophils, as you're preparing, they're dying, so it doesn't really get into the single cell suspension. And so the relative level of different, especially totally different immune cell types, we have to be cautious about what we conclude, because they may not have similar level of, or they may not be easily made into the single cell sample preparation. Also, it is good to compare the differential expression of different cell clusters in the same sample. So for example, if in the same tumor, I see kind of a progenitor T cell and a exhausted T cell or dysfunctional T cell are those drunk T cells that are no longer killing the cancer cells. You can compare the expression of those two cell types in the same single cell data. But when you compare the differential expression of the same cell cluster across different samples, you need to be careful, because some people will just, process, two different tumors, say one is responding to immunotherapy and one is not responding to immunotherapy. You do single cell RNA-seq. When you overlay those data together, you might see differential expression. And because there are hundreds of cells in that cluster, you might even have a very significant p-value. But the reality is you only have one patient in group 1 and another patient in group 2. It's never going to give you a confident enough readout. And so be very, very careful when you are comparing, the differential expression of single cell across different samples. So make sure you still need to have biological replicates to really call differential expression, and also to call the differential cell populations. Okay, questions?